So now coming towards mitochondria. For the mitochondria, we have enzyme which is glutamate dehydrogenase enzyme, which is the marker for the mitochondria. So mitochondria are actually the powerhouses of the cell, which make the ATP. For mitochondria, we do this diagram always. It has an outer membrane and an inner membrane. The inner membrane has a large number of foldings, which are known as the cristae, and they enclose the mitochondrial matrix. These inner membrane mitochondrial cristae, they contain these large number of enfoldings which serve to increase the surface area, and inside the mitochondrial matrix we have oxidative and phosphorylating enzymes. Mitochondria, they are present in the cell's cytoplasm, but their number varies from 100 up to several thousand. It depends upon the cell's activity. The cardiac muscle cells, which are the cardiomyocytes, they require a large number of energy. They have a large number of the fat cells, as a result of which they have a large number of mitochondria. The length of the mitochondria is 1 micrometer in diameter and 7 micrometer in the longitudinal length. Mitochondria are self-replicative, and they are involved in the synthesis of the adenosine triphosphate, which is also known as the ATP, which is the energy currency of the cell. Mitochondria have their own DNA, which is a small circular DNA which resembles the DNA of the certain bacteria. Then they have their own ribosomes, they have their own hydrolytic enzymes. So mainly mitochondria are involved in the protein synthesis. They have their own metabolic machinery, and because they're self-replicating, so their replication is actually controlled by the DNA, small circular DNA, which was present in the mitochondrial matrix. Cells that are faced with increased energy demands, like skeletal muscle cells, in certain exercising muscles, the number of the mitochondria, they tend to increase, and their number is directly proportional with the activity of the cell. So now, to understand the process that are actually happening inside the mitochondria, we have this diagram. So formation of ATP occurs in the mitochondria. What happens that certain compounds like the carbohydrates, they are converted into glucose, fatty acids from the lipids, amino acids from the proteins, they are formed in the cell, and the glucose, it undergoes glycolysis and produces 2 ATP, and now this portion, it moves to the interior of the mitochondria in the form of pyruvic acid, whereas the fatty acids and the amino acid, they are converted into the acetoacetic acid, and now the pyruvic acid, the acetoacetic acid, they are converted into acetyl coenzyme A, which is the main form which takes part in the process of Krebs cycle. Here the process of Krebs cycle is produced, and here the process of Krebs cycle takes place, and the 36 ATP, ADP, they are converted into 36 ATP. Along with that, with the oxygen and carbon dioxide, they are released. Oxygen is taken in, carbon dioxide is released, and also water is released outside of the mitochondria, and this is the process through which we get the process of ATP formation, and this is known as the energy currency of the cell. This is why mitochondria is the energy currency of the cell, and this is known as the chemiosmotic synthesis of ATP. 95% of the process, it occurs inside the mitochondria. 5% of ATP production takes place outside by the process of glycolysis. So now we discuss the ATP molecule structure. Here we have a ribose sugar, to which we have the phosphate group attached, and here we have the nitrogenous base which is attached. It can be adenine, guanine, cytosine, thymine, anything. And the phosphate group, if it is 1, it is known as adenosine monophosphate. If it's 2, it's known as adenosine diphosphate. If there are 3, it is known as adenosine triphosphate. And if only a nitrogenous base and a ribose sugar is present, it is known as a nucleoside. It is converted into a nucleotide whenever a phosphate group is attached. Now this a little bit deviated line, it actually shows a high energy bond, and whenever this bond is broken down, it releases 12,000 calories of energy. And for the Formation of this bond, we also require 12,000 calories of energy, which comes from the food stuff, that is via the glucose, amino acids, and fatty acids. Now, this ATP, which is produced inside the cell, it is involved in a lot of functions. Firstly, it is involved in the functioning of muscle contraction. Then it is involved in the functioning of synthesis of chemical compounds, that is protein synthesis. Then it is involved in transport of substances through the membrane. The transport is of the sodium, the muscle contraction, the actin myosin cross bridges formation by the ATP, and protein synthesis also requires ATP, which are provided by the process of Krebs cycle occurring within the mitochondria. One important fun fact over here is that membrane transport function, like the function, like the transport of the ions across the cell, it is very much effective, or it occurs in large amount in the renal tubular cells. The renal tubular cells, which are involved in the sodium secretion or the absorption of the substances, and tubular filtration and tubular absorption, they use 80% of their cellular energy or ATP in the process of membrane transportation. And certain cells use 75% of all the ATP of their cells to produce compounds, that is the protein synthesis. Also, the ATP that is produced, it is involved in locomotion, that is either the ciliary locomotion or the amoeboid locomotion. So we have the synthesis of the compounds, the membrane transportation function, and muscle contraction. Along with that, we have the locomotion, which is either the amoeboid locomotion or the ciliary locomotion. So, mainly ATP is involved in all the active processes inside the cells. Now we have mitochondrial related diseases. One is the myoclonic epilepsy, ragged red fibers. It is epileptic disease. Then we have the mitochondrial encephalomyopathy, lactic acidosis, and stroke like episodes. It is also a mitochondrial degenerative disease in which there is lactic acidosis and a cardiac disorder. Then we have a neuronal disorder, which is neuropathy, ataxia retinitis pigmentosa. You just have to remember the names of the disease that are related to the mitochondria. So now we have another non-membrane bounded organelle, which is the cytoskeleton. It further has three parts, which are the microtubules, the intermediate filaments, and the microfilaments. The microtubules are 25 nanometers in diameter, and they contain alpha and beta tubulin. They actually act as conveyor belts for the transportation. They are involved in centriole, mitotic spindle, and formation of cilia and the cytoskeleton. Intermediate filaments, they are intermediate in size between the microtubules and the microfilaments. They are 8 to 12 nanometers in diameter. They contain fibrous protein. They are involved in anchoring function. Since they anchor, they provide mechanical support, but they are less dynamic. They do not move like the microtubules. Then we have the microfilaments, which are 7 nanometers in diameter. They contain G actin molecules, then they contain F actin molecules, then they have two intertwisted F actin chains and the tropomyosin and troponin, etc. They provide elastic support, they do the recoiling process, and they are involved in ectoplasm formation. Here you can clearly see the microfilaments, which consist of two intertwisted F actin chains and the single molecule. These are known as the G actin monomers. Then we have the microtubule. Main function is to function as a conveyor belt, and it is made up of alpha and beta tubulin molecules. Then we have these intermediate filaments, which are actually supporting roles, and here you can easily see how they support the mitochondria with the microtubules. So that is very much important. Moving towards nucleus, the main enzyme marker for nucleus are RNA polymerase and DNA polymerase, sometimes histone deacetylase. So now I will explain the nucleus through this diagram. So here we have a nucleus which is surrounded by a double membrane bounded membrane, which is the nuclear membrane, which is continuous with the endoplasmic reticulum. Now this fluid inside the nuclear membrane is known as the nucleoplasm, and a densely stained region which is the nucleolus. The nucleolus has further two parts, a central region and a peripheral region. The peripheral is fibrillar region and a central is the core. The outer membrane of the mitochondria is fused at various points. The two membranes are fused at various points, and the fusion points they actually are separate, and these are known as pores. And these pores are 9 nanometers in diameter and provide the transportation of the substances from the nucleoplasm into the cytoplasm and from the cytoplasm into the nucleoplasm. This nucleus it is present in the condensed form in dividing cells. In the dividing cell, the nucleus is not present either. It is present in the form of the chromosomes are present there. But the chromatin material, which is the uncondensed form, it is present in the nucleus in the interphase period. That is the non-dividing cell period. The main function of the nucleolus is ribosome formation. It actually performs the function of attaching the lower and higher ribosomal portions with each other, that are the 50s and the 40s ribosomal portion, which are attached in this area, and the ribosome move from the nucleolus into the nucleoplasm and from the nucleoplasm out of the nuclear pores into the cytoplasm. The nuclear pore, which is a 9 nanometer pore, it can also allow molecules weighing up to 44,000 to pass through. The enzymes that can move inside the nucleus, they are DNA polymerase and RNA polymerase, and the substances that move out, they are namely the ribosomes, DNA, and the messenger RNA, etc. The ribosomal subunits are also present in the nucleolus because it is involved in the formation of the ribosomes. Here we have the diseases related to the nucleus, that is xeroderma pigmentosa, in which we have the sun sensitivity. Then we have the laminopathies, in which there is mutation in intermediate filament synthesis. And then we have Emery-Dreifuss muscular dystrophy. It's a type of muscular dystrophy in which the muscles are not able to work, and the short form of this is EDMD. Now we have the comparison of various cells. The smallest virus is 15 nanometer, the large virus is 150 nanometer, bacteria is 350, and the smallest bacterium is Rickettsia. Normal bacteria is 1 micrometer, and cell is 5 to 10 micrometers in size. This is a size comparison of a prokaryotic versus eukaryotic cell, or non-cellular particle, then prokaryotic, and then eukaryotic cell. Now, the cellular ingestion method, we have endocytosis. Endocytosis is the taking in of the material. It is a process of active transport and it requires ATP. Normally, the methods of transportation, we will study all of those methods in chapter number 4, but we have mainly diffusion and active transport. Diffusion does not require ATP, it is simply a movement, whereas active transport requires ATP molecules, and the endocytosis process is cell ATP requiring process, and it is further divided into phagocytosis, that is ingestion of large particles, whereas pinocytosis means ingestions of minute particles in the form of vesicles. So we have this diagram which is enough to explain the process. Now here we have the cell membrane which has receptors attached to it, and it is a substance or the protein which is to be attached, and these are cladrin proteins. This receptors, they are located inside a coated pit. So now when the receptors, they have the protein molecules attached to them, we get a process of pinocytosis. What happens here is the cladrin molecules, they become activated. They start to form a vesicular structure. Actin and myosin, they appear at the edges. Now the dissolving cladrin, it actually converts the substances from the outside into an intracellular vesicle. And now we have the process of endocytosis completed. The limiting factors for this process are ATP, because it is an active transport process, and calcium ions, which are involved in the functioning of actin and myosin molecules. Phagocytosis also occurs in the same way, and it is just the dilution of the larger particles, but they have written it over here in a summarized manner. And also the attachment of the receptors with the protein molecules, it is known as the opsonization process. So, cell membrane receptors attach to the surface ligand of the particles. They can be surface ligands, they can be receptors. The receptors, they attach to the surface ligands, which can be proteins or other substances. Now, the edges, they evaginate outward and they form that clathrin area, that is the cup shaped area. Then actin and myosin appear, actin and contract fibrils appear, and the phagocytotic vesicle is pushed inside by the clathrin molecules. These contractile proteins pinch the stem of the vesicle, so vesicle completely separates from the cell membrane and it is moved to the interior, that is the cytoplasm, in which it can be either the phagocytotic or the pinocytotic vesicle, depending upon the amount of the material it has ingested. Now, the digestion of the phagocytotic or pinocytotic vesicle occurs by the enzyme that is derived by lysosome, because lysosome contains 40 different hydrolytic enzymes. So, here we had the process of opsonization, then movement inside, then dissolving, and then finally pinocytotic or phagocytotic vesicle has been formed. It is a primary vesicle. Now, lysosome fuses with it and forms a secondary or a digestive vesicle. After that, the substances which are useful, they are released into the cytoplasm, whereas the substances that are not useful or to be excreted out, they are converted into a residual body which moves out by the process of exocytosis. This was endocytosis, opposite of which is exocytosis. Now we come towards locomotion of the cells. Most obvious type of locomotion, it occurs by muscles, which are the skeletal muscle, cardiac muscles, smooth muscle cells. They constitute entire 50% of the body mass. Now, two other type of the movement, they are amoeboid movement and ciliary movement. For the amoeboid movement, it is a crawling like movement. And the main substances that perform the amoeboid movement, they are the white blood cells, that are macrophages, the germinal cells of the skin, fibroblast cells, and developing embryological cells. I will explain the process of amoeboid movement via this diagram. Actually, what happens is at the that this amoeboid movement occurs by the process of pseudopodium formation. What is a pseudopodium formation? What happens is that at the moving end, chemotactic substances or the substances that cause the motion, they cause the release of certain vesicles from the cell. As a result, these vesicles are released by exocytosis process. They bind to the receptors and they pull the molecule forward. Similarly, at the end opposite to the developing end, we have endocytosis. There is movement inside. These vesicles, they move outside, they form a pseudopodium. This pseudopodium then proliferates, it releases its receptor, they attach to the substance, and then it moves forward. Let me show you it via our animation. Now we will look at the process of locomotion over here. We can see in this diagram that the vesicles are formed and they move inside the cell. The previous portion shrinks. These vesicles move to the rear end and they release the substance outward and a pseudopodium is formed. This is how the cell moves from the rear end towards the leading end, like this way in the form of bricks. What is actually there is actin and myosin filaments which are moving or protruding into the pseudopodium, and they cause the pseudopodium to move forward and it moves in this direction. And now we see the whole of the cell in action, forming the vessels and the pseudopodium. And the amoeboid motion is actually controlled by the pseudopodium. And the pseudopodium occurs and moves via the chemotaxis process. Now these are the chemotactic particles. The cell moves towards the area where the chemotactic particle concentration is higher, forming its pseudopodium. So now moving towards an area of higher concentration of the chemotactic particles is positive chemotaxis, whereas moving towards an area of lower concentration of the chemotactic particles is negative chemotaxis, or away from the positive stimulus. And the main cells that do the amoeboid locomotion are white blood cells, the macrophages, the fibroblast cells, and the embryological cells, which are involved in the formation of the embryo. Let's look at a brief summary. So we have this endocytosis process, exocytosis, and pseudopodium formation by the motion of actin and myosin filaments due to the chemotaxic substances in the direction of the chemotactic substances, that is positive chemotaxis. So this is how the cilia are going to move. So now why did I showed you the cilia? Because we are going to discuss the ciliary motion, the mechanism of ciliary motion. Now mainly first we have to discover what a cilia is. This is the tip of the cilia. This is the body of the cilia, which has a filament surrounded on the outer side by a membrane. It has a basal plate. Then we have a basal body and a rootlet. The cross section is over there. It consists of 9 peripheral duplets and 2 central singlets. So now we are going to study the structure of cilia from here. So this is the structure of ciliary axoneme, which consists of 2 single in the center and 9 peripheral. In the 9 peripheral, we have this protofilament. This is a duplet. It has nexin and dynein arms. Then it has a radial spoke which is moving towards the interior. These are actually the dynein arms. So now in the functioning of cilia, we have 2 types of strokes. One is the forward stroke, other is the backward stroke. The forward stroke is also the powerful most stroke, and it is known as the effective stroke. It is mainly known as the effective stroke, which is the forward stroke, and the backward stroke is not the effective stroke, and it is mainly formed by, if the forward stroke is done by 5 cilia, the backward stroke will be done by the 4. Now here we have an overview of this cilia and its transportation. So cilia are motile and they can also be non-motile, which are known as the primary cilia. The movement of cilia is whip-like movement, whereas the movement of the amoeba was crawling motion. They're present in respiratory airways and in the uterine tubes. Uterine tubes for the movement of the egg inside the uterus, and the respiratory airway for the movement of mucociliary transportation. The movement rate of the ciliary transport is 1 centimeter per minute towards the pharynx, that is mucociliary transportation. And from the ovary to the uterus, this movement is known as the mucociliary. This movement is known as the uterine movement. So on the surface of the cells, there are 200 cilia and they are 2 to 4 micrometers in diameter. The main thing is that they have a basal body and a rootlet, which we have seen over there. And from the basal body and the rootlet, a filament grows, which is surrounded by a main thing, which is the cell membrane. Now, flagellum of a sperm, it is also a motile cilium, but the movement it does is not a whip-like movement. Rather, it is a quasinocidal waves. The quasinocidal waves are actually, for example, this is a sperm. So the sperm's tail will move in this motion. This is a quasinocidal waves. More like quasi-sinusoidal waves. We've also done the structure of the axoneme, which consisted of 9 peripheral duplets and 2 central singlets. The main action what actually happens is that the central singlets, they receive the main action data and they transmit it via the radial spoke to these peripheral ones which interact with each other and crawl on each other via the dynein arm, as a result of which we have a whip-like motion of the cilia. The movement of cilia depends upon the availability of ATP because it is a active movement. Ionic condition, that is the balance of calcium and magnesium ions, and multiple dynein ions that are involved in this process. The dynein arm of the cilia, they are actually adenosine triphosphatase ATPase arms which use the ATP, do the enzymatic reaction, and then they perform the function. The movement, it depends upon signals which are the electrochemical signals. These come to the central tubules and they activate the dynein arm. Sorry, I was not showing it. So they move to these electrochemical signals, they are transmitted via the two central tubules to activate the dynein arm of the peripheral ones. Now certain cilia are there which cannot even move, so they are involved in sensations and signal transmission as well as cell growth. They are actually sensory antennas of the cell. They are large in number but small in length. These primary cilia, they are very much important because they are non-motile and they do not contain the dynein ATPase arms. But there is certain defects which occur in the primary cilia, and due to these defects we have polycystic kidneys. Poly means many, cystic are cysts or abnormal aggregates. Kidney is kidney or renal area. So large fluid filled cysts are formed in the kidneys due to the abnormal functioning of the cilia in the renal tubular epithelial cells. Sometimes we also have a difference between lysosomes and peroxisomes. Firstly, you can write the enzyme markers. For the lysosomes, it was acid phosphatases. For peroxisome, it was uric acid oxidase. Lysosomes, they are formed by Golgi apparatus. Peroxisome formed by smooth endoplasmic reticulum, also Golgi apparatus. The main enzymes the lysosome have, they are hydrolytic enzyme, whereas peroxisome have oxidative or peroxidative enzymes. Lysosomes do the function of intracellular digestion, autophagy, and autolysis, whereas peroxisomes do detoxification. Lysosomes, they are larger in diameter and size, whereas peroxisomes, they are smaller in diameter and size, and they have different disease. Peroxisomes have peroxisome bar degenerative disorders and Zellweger syndrome, whereas lysosomes have lysosomal storage diseases like Tay-Sachs disease and Gaucher's disease. I hope you understood each and everything. Let's meet in the next chapter. Till then, Allah Hafiz.